So, good morning, good afternoon, good evening, everyone, and welcome to today's webinar. This time, the very first webinar of a new series organized together with the Plant Journal. My name is Isabel Mendoza. I'm delighted to welcome you on behalf of the organizing team. And before we begin, a quick reminder about some of the how this webinar is going to work. As you know, it's being recorded and will be available afterwards on our YouTube channel. Please feel free to share with your colleagues who may not be able to join today. Plus, the Q&A session will happen at the very end of the session, and send your questions through the Q&A tool on the toolbar on your Zoom screen. And now I will give the floor to Katherine and Denby, Editor-in-Chief of the Plant Journal, who will be also moderating today. Katherine, the floor is yours.
Great, thanks Isabel. And yeah, welcome everyone to our first Global Plant Council and Plant Journal webinar. It's great to have so many attending. So the aim of the webinars is really to sort of enable scientists to connect and also help our authors to disseminate their work and highlight their work. So we're going to have 4 Plant Journal webinars over the next sort of 10 months or so. And Global Plant Council are also organising webinars with the Plant Biotechnology Journal, so we'll alternate. So for this first webinar, we've chosen 3 authors who contributed to our recent special issue on plant engineering biology, and we'll put a link to the special issue in the chat if you want to follow up with that afterwards. So the Plant Journal does an annual special issue, just one a year, comprised mostly of review articles on a topic. And this year's was about different technologies for plant engineering, both for the transformation process and design of strategies. So our speakers today contributed review articles to the special issue, but in this webinar, they'll be talking about their own research in these areas. So we'll have 3 20-minute talks, one from each of the speakers. And as Isabel says, we'll then have the question and answers at the end of those talks. And obviously, you can direct your questions to any of the speakers. And just to remind you again that please put your questions through the Q&A facility on the webinar, not the chat. Okay, so yeah, with that, I'm delighted to introduce our first speaker. So our first speaker is Keunsub Lee. He's a research scientist and adjunct assistant professor in the Department of Agronomy at Iowa State University. And his research focuses on tools for crop genetic transformation and genome engineering. He's engineering Agrobacterium strains and using new technologies to make transformation genotype independent and also available for a wider range of crop species. So over to you, Keunsub, and we look forward to your talk.
Thank you, Katherine and Isabel, for your invitation. I'm so happy to share our research. Today's topic is enhancing Agrobacterium-mediated plant transformation using improved ternary vector system and auxotrophic strains. So today's outline, I'll give you a brief introduction to Agrobacterium and Agrobacterium-mediated plant transformation and the different vector systems for agro. And then we'll go to the rapid B104 immature embryo transformation system, and then we'll summarize. So what you need for successful plant transformation? The first thing is you need to have a regenerable X-plant material, and second is the DNA delivery machinery. So in this case, today we are going to talk about efficient DNA delivery method using agro, but there are other methods available, but today's topic is agro. The last one is a regeneration. So you have to enrich the transformed cells and then regenerate fertile plant out of it. So this is not today's topic, but I think Greg's presentation will cover some of the regeneration. So this is Agrobacterium-mediated transformation, the process. What happens is the first plant cell, usually wounded cells, secrete some compound, including reducing sugars, phenolic compound. Let me change it to laser pointer, and then acidic pH. Then these are sensed by Agrobacterium two component systems, and that triggers the virulence gene expression that process the tDNA and then make the type 4 secretion system, which secretes the T strand along with the other effector proteins. So we understand, how different effector proteins are involved in the processing of tDNA and transport to the plant cells. But still, there's still unknown things here, how that exactly T strands are targeted to the nucleus end and how they're naked and then integrate into the chromosome. So there are different vector systems for Agrobacterium-mediated transmission. The naturally existing systems, such as a TI or RI plasmid, you can find the VIR gene, VIR region, and the gene of interest. In other words, the tDNA region are located in the same DNA molecule. So this is a unitary vector system, okay? And in late 1970s, people discovered it. If you replace the tDNA, natural tDNA, using your gene of interest, you can transfer it into the plant cells. That, revolutionized bioengineering, and the people immediately interested using it as a tool for plant transformation. The problem is you have to introduce your gene of interest in a form of a co-integrate vector, which does not replicate within Agrobacterium, but should, go into this, tDNA region of your TI plasmid, which is not very easy. And then came the binary vector by splitting the T region into a separate plasmid so that it's more easier to manipulate. And then came ternary vector system. So I'll go over one by one. So unitary vector system is a natural system, but as I already mentioned, the cloning step is very, very cumbersome and inefficient because the co-integration process usually rely on antibiotic selections and then testing, whether the co-integration was precise or not, it's not easy. So therefore, early 1980s, when the binary vector system came out, it became immediately popular because, you can make the small binary vector, which is easy to manipulate, and then you can put on a different original replication or copy numbers so that you can have a higher copy number that may enhance the transformation efficiency. So this is definitely, compared to the unitary vector system, much more, amenable and easy to manipulate. However, it was not great for monocut transformation because it was not until 1996 when the Japan Tobacco developed the super binary vector system, which contains about 15 kb fragment. It's called a Komari fragment, which has a via B, C, G operand from the super binary, super hypervirulent Agrobacterium strain, in this case, the PTI-BO542. And it really enhanced the transformation efficiency in monocut, including rice and maize. So after that, many people have been using this one. But again, the downside of this super binary vector system is that you still use the same co-integration process to clone your gene of interest because of the larger size of the super binary vector. So that was the downside. But because at the time, monocut transformation was so inefficient, super binary vector was still popular. Then came the ternary vector system. Now we realize that, if we have an extra copy of a virulence genes, you can significantly enhance Agrobacterium-mediated transformation efficiency. What if we split the super binary vector into two pieces? That came the ternary vector system. So the first tDNA delivery machinery of VIR genes provided by disarmed TI plasmid. Now we have an additional plasmid. And as I mentioned before, the copy number of a plasmid play important role because there is a definitely dosage effect. TI plasmid is usually a single copy before the virulence gene induction, but upon induction, the copy number may increase. However, the helper plasmid, depending on the origin of replication or copy number machinery, you may have a much higher copy number, such as a PBS1 or RK2. It has about 7 to 10 copies per cell. So therefore, you already have a much higher copy number to start the virulent gene induction. Therefore, it gives you a boost for transmission. And then it also takes advantage of the binary vector system because your binary vector, tDNA vector, is on a small original replication, so you can easily manipulate it. The only thing you have to check is that it requires plasmid compatibility, meaning that your Agrobacterium strain itself, some of them have an existing antibiotic resistance gene, and your plasmid, especially the helper plasmid and your binary vector, they must have a compatible original replication. In other words, you should not have two plasmid with the same original replication. That's the bottom line. And also the antibiotic resistance gene, that you need to have a different antibiotic resistance gene. Ternary vector system. We use this for immature embryo transmission for maize B104. We are using thymidine auxotrophic Agrobacterium strains. I don't have an extra slide for auxotrophic strains, but here I will just explain briefly. Auxotrophic strains are developed to minimize the overgrowth during tissue culture. So in this case, we knock out the Thi-A gene in Agrobacterium. Therefore, you need to supplement thymidine into the media. Without it, Agrobacterium cannot grow. So you only add it until co-cultivation period. After that, you stop adding it, and then Agrobacterium overgrowth can be controlled. Another thing is the biosafety concern, containment, because these days we are using a lot of recombinant DNA, including CRISPR reagent. And if agro is auxotrophic, they are much less likely to survive in the outside of laboratory conditions. So this is another safety measure. And our helper plasmid, I made it based on the RK2, which is a broad host range plasmid. And it's also medium copy number in Agrobacterium cell. So I have two helper plasmid, 2299 and 2299A. Both of them have a gentamicin as a selection. And our tDNA vectors, I'm using PVS1. This is also medium copy number, about 7 to 10 copies per cell. And then I use spectinomycin or kanamycin, and depending on the Agrobacterium strain. And these are the maps for helper plasmid. Both of the helper plasmid are available from Adagene. One thing I want to point out is that you have to check, I just emphasize again, RK2 original replication is compatible with any tDNA vectors, which has a PVS1, PRI1, PSA original replications. You can freely mix and match as long as the antibiotic resistance genes are not identical. For today's talk, I used two binary vectors. One is a CRISPR-Cas9 construct with mCherry as a selectable marker. We switched to MPT2 or G4118 selection for plant because of a lot of escape issues with bar selection. Another binary vector, we used the Ruby, which is a newly developed visible marker gene. I'll show you a little bit more detail in the next slide. And again, same MPT2 selections. So Ruby was developed by Professor Yunde Zhao's group at UC San Diego. They cloned 3 essential genes for biosynthesis of the purple pigment called bethylene. You can see this color in some flowers and a bead. It converted tyrosine into this colorful pigment. And then it gives a really vivid color during tissue culture. So we just started using it in 2021-ish. And then this is the one day after co-cultivation with immature embryo. So you can see the red or purple colorations. It indicates that tDNA was successfully delivered and they were expressed within immature embryo. And the tissue culture, callus stages, or shooting stages, or even rooted plantlets, you can visually monitor the transmission process. So we really like the Ruby reporter for transmission. And then I talked about that helper plasmid enhances tDNA delivery efficiency. How much, right? So we use the Benthamiana agroinfiltration system, one strain without helper, here A2, and the one strain with a PKL229 helper plasmid. You can, if you look at the leaves after infiltration, you can see the different pigmentation levels, and we quantify it, and we saw that there's about 3 to 4-fold increase compared to the control. So without helper plasmid, it was around 129 milligram per kilogram fresh tissue. It was boosted about 3 to 4-fold with helper plasmid. So it really shows that the helper plasmid enhances tDNA delivery efficiency. Now, how does it work for a stable maize transformation? So we developed this rapid B104 immature transformation. Our conventional method took about 5 to 6 months. And it starts with the same immature embryo, which is about 1.8 to 2 millimeter. And after Agrobacterium infection, co-cultivation, the old method utilizes callus induction medium, and then proliferation and bulking stage, which takes about 4 to 5 months. And the actually regeneration process takes about 4 weeks. It's the same, but the difference is whether we are going through this callus induction and bulking stage, or we immediately go through somatic embryogenesis and the regeneration process. So the current protocol we use is the same immature embryo because it's regenerable. After overnight co-cultivation, we immediately put them on the resting media. There's no selection yet. About a week after that, we put them maturation media. Here, the somatic embryos mature. After that, we put them onto the regeneration media, which takes about 4 weeks. So the overall process takes about 7 to 8 weeks. You already have a rooted maize transgenic plant. So this is illustrations, how we process. You have to isolate these immature embryos, right? And then we collect them in small tubes. After that, you do the agro-infection for 5 minutes, and then you put on the cultivation medium. So the key point is that you have to put the embryo axis, which is flat side of the immature embryo, facing the medium, and the scutellum side should face the top. And after that, you can immediately see transient expression of reportogen. For the CRISPR-Cas9, you can see the M-cherry expression. And but with the Ruby, you don't need any special tools, or you don't have to sacrifice your tissue to visualize the transient expression. So after maturation, we put them to the shooting stage, and you can see transgenic plant with the Ruby pigmentations. So far, it has been working really, really nice. Now I'm going to show you some very, very interesting result. So we compared different Agrobacterium strains and without helper plasmid. For instance, we have been using EHA105 strain, EHA105 Thi- for maize transformation for such a long time. Well, with this rapid method, without helper plasmid, you don't get anything, okay? And then we compare this one, LBA44 for Thi-, and without helper plasmid, again, we repeated this experiment multiple times, and we cannot get anything. So our old protocol utilized 3 to 4 day co-cultivation with using regular binary vector. We were getting about 4% efficiency. But with this rapid method, only overnight co-cultivation, without ternary helper, there's nothing, literally. But with this new helper, we are getting average about 55%. With the old helper, with the CRISPR-Cas9 construct, you expect a lower efficiency, but it's mainly affected by immature embryo quality. So when immature embryo is really good, you get really, really high transformation frequency, near, close to 100%. But when they are not good, still with the helper plasmid, you can still retrieve about 10% of a transgenic plant. Then we did a side-by-side comparison, with the new helper and old helper. The only difference is the new helper, I cloned the VIR-A gene. So from same origin, AGL1 TI plasmid. And you can see that immature embryo, number of immature embryo, and the immature embryo with the Ruby, transient Ruby expression, and this is a percentage. What I want to point out is that within experiment, the new helper with the VRA gene has a higher transformation frequency or higher transient Ruby expression in all 3 experiments. However, transient Ruby expression itself is not a good indicator. For instance, the first experiment, nearly over 90% of them have a Ruby expression. However, the regeneration was the lowest. The third experiment, on the other hand, has a very, very low, poor Ruby expression, but in the end, you have a medium level of transmission frequency. So this means that Agrobacterium, the tDNA vector system, really enhances the tDNA delivery. However, the regeneration cannot be solved by this regular Agrobacterium strain with just ternary vector system, but it does enhance tDNA delivery in efficiency. I think I went a little bit too fast, but I think we saved some time for our discussion after the talk. Here are the 3 take-home messages. First, the ternary vector system with additional virulence genes will significantly enhance the tDNA delivery efficiency. If you want to implement ternary vector system, make sure that your helper plasmid is compatible with your existing binary vector system, also Agrobacterium strains. You don't want to have a same bacterial selection system, such as antibiotic resistance genes, or same original replications between the ternary vector system, tDNA vector system, and your helper plasmid. Lastly, I want to point out that Agrobacterium does not resolve the regeneration problem in most cases. I think the technology is coming up. I think hopefully new engineered strains can also contribute to the enhanced regeneration. I think that it'll come hopefully soon. With that, I'd like to thank my group who contributed this work, my supervisor, Professor Kan Wang, and my lab members, and especially the B104 rapid transformation method was developed by our former student, MJ Kang, and with a collaboration with Dr. Vina from Danforth Plant Science Center. And also the crop bioengineering laboratory, Dr. Jessica G and Sehiza, they did most of the transformation. And the helper plasmid comparison experiment was done by Anna as a visiting scientist. Thank you for your attention. And I'll stop here.
Great. Thanks, Keunsub. That was great. And there's already some questions coming up. So just to remind everyone, put your questions in the Q&A facility. I'm sure there'll be many more. Brilliant. It was very clear and really, yeah, exciting. Okay, our next speaker is Greg Goralogia, sorry, who is a research associate in the group of Stephen Strauss at Oregon State University. Greg's really focused on understanding transformation and regeneration in trees, and then using that knowledge to develop improved regeneration technologies. And he also uses sort of gene editing tools for engineering forest trees for various applied traits and fundamental biology. So Greg, if you want to start your talk, thank you.
Wonderful. Thank you, Katherine, and Plant Journal for this wonderful opportunity to share some of my research. So today I'm going to talk about this transformation system that we developed that leverages genes from the Agrobacterium rhizogenes for hairy root culture. So in our group, we work mostly in trees, and that includes predominantly poplars that are members of the genus Populus. Here I'm showing you hybrid poplars, but we also work in some really challenging species like eucalypt hybrids. To my knowledge, this is the first approved field trial for CRISPR-edited trees in the US. They are now in their 8th year in the field, so it's kind of a career-spanning length of time. These are edited for some sterility traits, and actually this is the first year that we've gotten some flowering from some of these, so we can actually phenotype them. So it's quite a long process. And as I share this transformation system, that's kind of the perspective that I come from. So we're talking about long testing cycles, some regulatory challenges, and most of these are also interspecies crosses that come from really long breeding programs. So the breeders want traits in very specific clones, and that provides a lot of challenges for biotech. So in general, these species are also just really challenging in terms of the in vitro culture techniques. So again, these are elite clones. We don't have access to juvenile material like seedlings. Mostly they're very heterozygous, so every genotype is kind of a new adventure, and there's a lot of physiological diversity in the explants. Eucalyptus is kind of the worst that we deal with. Some petri dishes shown here. This is actually the best performing of about 100 genotypes we screened. And you can see when you add antibiotics as well as agro plus antibiotics, things kind of go haywire. And there's a lot of necrotic tissue and strong immune responses. So if we think back on the last 30 years, kind of conventional plant transformation techniques, we need some kind of a DNA delivery agent, in this case, predominantly Agrobacterium. And then we're going to use probably phytohormones mostly in the medium in order to exert developmental effects. And so those are coming exogenously in the tissue culture media, and we're going to use those to form callus and shoots predominantly. In the last couple of years in particular, people have thought about using morphogens, so genes that are included as part of the tDNA, and those are going to exert developmental outcomes upon transformed tissues. In some cases, these have varying degrees of cell autonomous versus non-cell autonomous effects. And those are going to behave in both similar and different ways to how we might use phytohormones in tissue culture media. So we've used a lot of these in tree species. You may be familiar with, for instance, the Wuschel baby boom system from Corteva in maize, but also GRF-GIF has been quite popular across monocots in particular. And for the most part, we really have found these not to be working very well in woody dicot species. So we've tested things like Wuschel baby boom, GRF-GIF, LEC1, LEC2. And so predominantly, we've focused on using genes from Agrobacterium that seem to be powerful in terms of their developmental effects in woody dicot species. So one of the most important sets of genes from Agrobacterium is ones that cause hairy root disease. Here's a picture of these in a hydroponic tomato. They cause root proliferation, and they're from a very specialized clade of Agrobacterium. They tend to be underrepresented in wild strain collections, but a lot of them have been used for almost 30 years in terms of this culture technique, which relies on roots rather than callus and shoots. Hairy root culture, again, produces these transgenic roots, and it's actually remarkably effective in a wide diversity of plant species. So, I work in trees, and some of the most difficult ones, including some gymnosperms, are amenable to this technique. Things like larches, ginkgo, eucalyptus, which I've said is very challenging, radiata pine, as well as willows are quite recalcitrant. And a lot of the transformation systems that I develop are also quite relevant for specialty crops that are clonally propagated, things like sweet cherry and grape that are amenable to this technique, but very, very challenging to use a disarmed Agrobacterium strain. So hairy root transformation has become kind of a widely used technique because it's so effective in some of these species. So for instance, this is a study that was published a couple of years ago in citrus, where you might generate these hairy roots, but then use in vitro cell culture techniques in order to regenerate whole plants from them. The challenge with this, of course, is that in some of these regenerants, the hairy root-inducing genes might cause some pleiotropic effects. Occasionally, they're silenced, but oftentimes these transgenics have dwarfism, architecture phenotypes, change floral morphology. And so, if you have the right controls, this could be useful in non-model systems, but there are these kind of negatives for this approach. So we had a concept that we term reset, in which we might use these hairy root genes as morphogens in plant transformation. And so it might look something like this. We use Agrobacterium, and we're going to use genes from these Agrobacterium rhizogeny strains in order to produce transgenic hairy roots in culture. Then we might use morphogens that help induce shoots from those root tissues, or in addition, potentially use phytohormones in the media to spur this process along. Then we need some method to actually remove these genes somatically from these tissues and regenerate whole plants out of that. From there, we might be able to see whether we have any gene edits or whether we've successfully removed the transgene. And the benefit of this system is that we might be able to retransform the same clone again, which in terms of engineering would be advantageous in a lot of these clonally propagated species, and from there move on into the field. So the concept that I shared with you is not just my own idea. So Hirayasu Ebenuma, then at Nippon Paper Company, had shared some of these ideas in the late 90s and early 2000s. Some of the vectors that they've developed there were frankly amazing, but the cloning technology of the time really inhibited their general use. And they started to assay whether these transformation strategies could work. So let's build this thing. So this is going to be pretty complicated. So if we look at the hairy root inducing genes, type 1s like K599, which is a common strain isolated from cucumber, the native tDNA looks like this. And the primary gene that causes the hairy root proliferation is this ROLB gene that's located within this cluster. And then for woody plant species, these type 3 RI plasmids are the most common. They typically deliver 2 tDNAs, including the one that's similar to K599 like I showed you, but also some auxin-inducing genes, as well as a second copy of this ROLB gene. Based on some work in the literature in terms of how these affect, we chose a 6-gene cluster from the widely characterized strain A4 for use in this workflow. And in terms of shoot induction, we chose a couple of morphogens. And so Wuschel has been shown to aid the transdifferentiation of shoots from root tissue. So we chose that. We also used the Agrobacterium IPT gene, which produces cytokinins. And we expressed each of these under heat shock promoters. We know these are leaky, but at least the 2 promoters we picked are validated in Poplar, our model organism of choice. And then for transgene excision, we used a Cre recombinase. So the whole transgene gets flanked by lock sites. And then at the borders of those lock sites, we also included some matrix attachment regions to help with transgene silencing and stable expression. So I should mention, and I don't want to scare you with the size of this construct, that this would really be impossible through conventional cloning techniques, or at least very, very challenging. So as Keunsub showed, typically people are going to be using binary vectors and a disarmed Vir plasmid. We use this in vivo recombination system called gantry, where we're actually able to do Golden Gate assembly, but then load those iteratively as cargo into the Vir plasmid. So this is actually more similar to the unitary system that Keunsub shared. So this is the whole construct. This tDNA is about 40 kilobases in size and includes 16 independent peptides. We also included 3 independent marker genes, so GFP, DSRED, and Ruby, like Keunsub talked about. That helps us to figure out whether the whole transgene made it in, and also gene editing components. So we targeted part of the gibberellin signaling pathway. We also included a hygromycin resistance gene, but for what I'm going to show you, we didn't apply any selection. So does it work? We tried this in 2 poplar genotypes, and here's an indication of what happens. You see this nice proliferation of ruby roots, in this case from leaf explants. It varied quite a bit based on the experiment, but generally between 25 and 50% of explants regenerated these hairy roots. And then we worked to try and figure out how we were going to induce shoots from the system. So we tested a couple of different heat shock durations. That's what's turning on the shoot and excision genes. And we found that about a 2-week heat shock period with a mild 39-degree pulse 4 hours per day was about right. And you can see those shoots shown at the right. This is a little bit perplexing, but when we added a short duration of heat shock, we actually saw a worse developmental outcome. We're not entirely sure about this, but 2 weeks ended up being about right. And many of the explants showed more than 10 shoots per explant, so we were quite happy with this result. And we have these multiple reporters in here, so we could go under the scope and then look for things where that whole giant transgene might have removed itself. So you can see these subtending ruby tissues, and as the shoots start to form, you can identify ones that have lost both the ruby and the fluorescent marker gene expression. So in this panel, you can see those potentially off at the right. Now, I will say that, we're using a recombinase, so there are dangers in terms of chimeric lineages of cells. And so it typically takes a while for these to sort themselves out. This is what they look like in later development. So you can see there's a mixture of ruby and visibly green shoots at the later stages of propagation. And we can clip off those green shoots and further propagate them in jars. In general, they look normal. We haven't done any greenhouse trials yet, but you can see the transgenics, even though there's a lot of condensation, have this kind of dwarfish effect, and you can still see the ruby expression. But one thing that we wanted to do was really further study the behavior of these in terms of the morphogens as well as the excision process. So we repeated these experiments and then isolated very particular insertion events, and that's the data that I'll show you from here on out. So one pesky thing about this system and sort of the biggest hurdle that we've had to overcome is that the transgene excision has a lot of problems with it, and we've struggled to get this to work, and we're happy it finally is, but it has this propensity to methylate transgenes in between the lock sites. So this is data from a new phytologist paper in 2021. You can see that in cells that express Cre, there's often a DNA methylation response between these. And so one of the things that I've been working on is trying to figure out ways to improve this excision process. And we have been working a lot on alternative lock sites that don't contain any cytosines within the crossover sequence to try and prohibit that failure to recombine. And in some constructs, that's been performing very well. And so I'll show you, this is basically the biggest effect that we've seen. And so I'll present those where we compare a regular LOX-P site against one of these customized lock sites. So if we look at the regeneration of shoots, comparing these two independent lock sites, you can see that in the LOX-P flanked construct, we get higher excised, visibly excised shoot production, as well as higher ruby shoot induction. So there's actually a regeneration effect there. We looked at where those subtending tissues came from, and basically the type of explant, whether it contained a pre-existing meristem or not, didn't affect the regeneration outcome. And we took these on for further gene editing and gene excision analysis. So in order to figure out what happened during the excision process, we use PCR. We have a couple of amplicons spanning the transgene. And again, this is collapsing into from this giant transgene into a very small footprint scar. And so we have a couple of primers spanning that region. The presence of any transgene band, we didn't categorize it as excised. And then we included multiple amplicons in the footprint in case there was a left border truncation. So if we look overall using these canonical LOX-P sites, most of the shoots and events that we obtained were fully excised, although we did have some underlying escape chimerism, so things that were not transgenic at all in the beginning and not edited. And in our alternative lock site, we generally saw the same trends where there were less excised, but also more escapes present in the population. If we look within events, you can see that for the LOX-P containing construct, 4 of the 5 were fully excised, and then 1 was just completely wild type escapes. And then again, for this alternative lock site, a little more complicated, but quite a few fully excised events that were present. So we looked to see if these were edited at all. Again, we were editing part of the GA signaling pathway, and we got quite a few edits so far. Some of this work is ongoing, but the editing rates seem to be between 50 and 70%. We've also been curious about these footprints, where they end up in the genome and what happens after excision. So we've used tail PCR and now completed whole genome sequencing on some of these events. In this case, we have the footprint that remains in a promoter of a transcription factor, but the insertion site is quite clean, actually, with just a 15 base pair deletion in that promoter region. But we also found events where, for instance, here we inserted the footprint into exon 1. This is a predicted knockout in one allele. But again, the excision process was remarkably clean. So most countries are going to regulate any gene-edited products that we do here as transgenics because there is an inserted piece of DNA. Practically, the smallest size of this could be is about 150 base pairs, but probably more like 200. So one of the things that we're doing is looking what is the consequence of this, especially in a clonal crop. So for some of these events we've characterized, does this impact neighboring gene expression? Or are there very low abundance transgenes that are still present in some of these clones? So that's sort of ongoing work that we're doing. Now, the real question, of course, is I showed you all this in hybrid poplar, which is quite amenable. Does it actually work in really challenging species? So we started to do some of this work in eucalyptus. We can get composite plants to form with these ruby hairy roots. In order to get this to work, we do have to add a few of these rhizogenic factors. So I showed you another tDNA that has this auxin synthesis gene, and that really seems to help with the root formation here. So we're trying to regenerate these and see if this is a viable platform in some of these really, really challenging species. So hopefully I've showed you today that this reset system that uses hairy root inducing genes can be useful in these clonal crop species. It's sort of a genetic regeneration circuit, if you will. We found really high rates of editing in these clones. One negative is that we did see quite a few escape shoots that were present, but we think applying selection in the right way could eliminate those in the early culturing process. And we hope that this will be a widely useful system in clonal woody plants where it's really difficult. We have no way to remove the CRISPR normally unless you go with a completely transgene-free approach. And where we're really thinking about this, and this sort of lends itself to the long testing cycle thing, is this system would be useful for simultaneous editing and trans or cis gene insertion. So you could imagine this construct being able to induce an edit in trans, but also leave a transgene. And for some of our species, that might be advantageous because the testing cycle is just so long. And so when we're done, maybe we just have stacked traits that are part of a single locus that can be introgressed into a breeding pipeline. So we think that this is kind of a versatile system for doing all of the above. So with that, I'd like to thank some people involved with the project. Victoria and Sydney have been the primary undergraduates helping me on this work. Here's Sydney and Carter, who's done a lot of the cloning on this in the field a week or two ago. We have a bunch of really talented tissue culture staff, including Kathleen, Kate, Parker, and Colette. And our boss, Steve Strauss, who's the tree engineer, has been very supportive of this work. This has been funded by NSF and USDA here in the US, and I'd be happy to take any questions. Thank you very much.
Great, thanks, Greg. That was great. That's yeah, quite some cloning there. But I'm sure people are going to be interested to hear it. Yeah, have questions around that. That would be really good. Okay, so just to sort of say again, it's great. There's lots of questions coming in. We're going to do the questions and answers after the third talk. So hang in there, and your questions will be answered. So now for me, it's a pleasure to introduce our third speaker, Chris Willig. So Chris is a postdoctoral fellow, also in the College of Forestry at Oregon State University. He does a lot of work on gene editing technologies in hop breeding and developing more flavorful and sustainable hop cultivars, but also has a fellowship which is working on the development of novel Agrobacterium strains for plant transformation. So a very complimentary talk to Keunsub that we heard earlier. So Chris, over to you. That was okay. And you also just need to take yourself off mute.
Okay, I'm off mute now, right?
Yeah, but you just need to be in
Okay.
presenter mode for your slides.
Yes. Okay, thank you, Katherine, and thanks, Isabel, for organizing this. As Katherine mentioned, a lot of this is going to be pretty complementary to what Keunsub presented and a little bit of overlap with Greg's talk, but I kind of want to give an overview of Agrobacterium strain enhancement generally, and then I'll get a little bit into the work that we're doing in our laboratory to generate novel strains for improving transformation. So kind of a roadmap of what I'm going to talk about. First, kind of our lab's journey to why we became interested in this topic, and then just kind of a broader rationale in plant biotechnology for why engineer Agrobacterium. I'll go over a little bit of the genomic organization of Agrobacterium. We'll talk about some of the target genes or sequences that have been used in strain engineering in the literature, and then some of the new old and some of the new tools that are coming out for accomplishing strain engineering. And then, as I said, a little bit about our work. So, as Greg mentioned, that we both work in the same lab, which was formerly called the Forest Biotechnology Lab, so very tree focused. I came into this lab working on hops, a specialty Pacific Northwest crop. And for a couple of tree species, Greg mentioned eucalyptus as well as hops, we can get in tissue culture by changing the plant growth regulators pretty good rates of regeneration. But when we add in transformation with Agrobacterium along with selection, we just get very low rates of either tDNA delivery or maintenance. It's kind of hard to tear those apart. In hops, we think that the problem is kind of the tissue that gives rise to regenerable shoots is just not getting transformed. In eucalyptus, as Greg showed, there's kind of the strong necrotic response going on when we expose the tissue to Agrobacterium. So this got us into thinking about exploring methods to boost tDNA transfer by working on the Agrobacterium. So generally, 95% of the literature publications that come out with new plant transformation methods make use of a small handful of biotechnology strains that we can think of as the conventional toolkit. So the ones I have circled in red here, this is kind of a pedigree of these strains. And anyone who works on plant transformation is probably very familiar with most of these strains. But we can see kind of how they originated. The wild type or oncogenic strains are relatively genetically constrained. Over on the right here, we have this phylogenetic tree that was published in the review paper in the plant journal that I co-authored with Greg. But this is kind of reconstituted phylogenetic data from a paper in Science published by Alex Weisberg, also at OSU, where she sequenced all of these these wild type strains we had in a collection, and there's a lot of diversity with respect to the virulence plasmids as well as the chromosomal background that we're just not making use of for plant transformation. So we're positing that by sampling from these more diverse background, we can actually we can make some kind of significant gain in plant transformation, especially in recalcitrant plants. So common traits that people have often sought to engineer agrobacterium for in the past. First, of course, the basic transition of taking what's normally a pathogen in the wild and making it into a useful vector for tDNA delivery in biotechnology, enhancing function of the DNA transfer machinery, such as the ternary vectors Keunsub discussed, increasing the stability of the construct for plant transformation within the agrobacterium cells, containment of agrobacterium growth, and then improving the tDNA insertion event copy number and quality of the specific integration events. Before I get too much further, I want to shout out this excellent review paper that was published by researchers 4 years ago who worked at KU Leuven and University of Ghent in Belgium. And I want to credit this paper with really kind of getting me seriously thinking about the possibilities as far as agrobacterium strain engineering. So to give an overview of the basic structure, again, I won't belabor this because Keunsub went into it kind of in depth. This is the structure of the strain C58, which is most widely studied. But I want to note that wild strains that have been sequenced kind of do deviate from this often. So we have a circular chromosome, a linear chromosome, technically called a chromid, a single accessory plasmid, and then all pathogenic strains are going to have virulence plasmid, which is either TI or RI that we name depending on the phenotype they induce. Other wild strains may have from 0 to 7 accessory plasmids with a lot of elements that we don't really know what they do. So disarmament is first, the transformation capability of the pathogenic strains depends on two elements present on the virulence plasmid. We have the tDNA region, which in a wild strain contains the oncogenes, and then the actual tDNA transfer machinery, which is in the vir region here at the bottom. And what disarmament is removing the native tDNA genes, so we no longer have a disease-causing vector, but retaining the DNA transfer machinery in the vir region. And then the most typical use case is a binary vector system where we decouple the tDNA onto two different molecules and deliver our own genes of interest. So what kinds of gene sequences have been targeted in agro strain engineering in the past? Endogenous genes. So REC-A is a kind of key gene in the DNA repair pathway of Agrobacterium that relies on homologous recombination. So strains that have had this gene knocked out are actually desirable for biotechnology because they offer increased stability of binary plasmids, particularly ones that use repeated expression elements, or if the transgene we want to deliver is like a large chunk of plant DNA, which often just does have repeat sequences in it. Again, won't go into this too much in depth, but auxotrophic mutants, as Keunsub said, they help contain Agrobacterium as kind of a biosafety concern, and then just practically in transformation, we want to eliminate growth of Agrobacterium following co-culture. Here's an example of plates that have really bad Agrobacterium overgrowth. And we can also possibly marginally increase transformation efficiency only because the methods we usually use to control overgrowth, heavy antibiotics and tissue washing, can be detrimental to the plant survival. Just a couple of other target genes besides the thymidine auxotroph that have been used. The thionine biosynthesis gene has been knocked out by a paper out of the Wayne Parrott lab, and then another paper used generated actually several auxotrophs in the AGL1 strain background, which have also been applied. So next, I want to talk about genes that we're adding into Agrobacterium, and in the review paper to kind of talk about these as all one thing, Greg and I came up with this kind of admittedly unwieldy acronym NTEEs, which stands for non-tDNA transformation enhancing elements. So the first kind of bin we put these NTEEs into is supplemental Vir genes, and this is precisely what Keunsub talked about. So we have the earlier use of the super binary plasmid system, which again relies on formation of a co-integrate vector, but importantly contains those supplemental Vir genes in addition to what's present on the native disarmed virulence plasmid. And then later on, we have generation of ternary plasmids, which contain usually a full complement, or Keunsub's recent iteration of this took this even further by adding the Vir-A gene to it. And in this configuration, of course, now we have 3 plasmids in our final cell, which the binary plasmid still carrying the tDNA for plant delivery. Another bin is what we've kind of grouped into what we call plant hormone modulating genes. So these consist of things like the transziatin synthase, or TZS gene, which is present in some Agrobacterium strains, the C58 included. It's absent from others. And this gene sequence is actually located on the virulence plasmid, but not in the tDNA region. So it catalyzes the production of a cytokinin, transziatin, which is thought to assist with the Vir gene expression and tDNA delivery. So previous research has shown when you clone this gene cassette and put it in strains that don't naturally express it, in some plant species, this enhances tDNA delivery. Others, it decreases it. Another example of hormone modulation is ethylene degrading enzyme, ACDS. So ethylene is a plant hormone, most known for being involved in fruit ripening and early maturity. And this research group in Japan showed that expressing this ACDS gene topically in Agrobacterium could improve melon and Arabidopsis stable transformation. GABA is another plant-derived hormone that negatively influences Agrobacterium by interrupting its quorum sensing function. And these researchers also showed that by expressing this GABT gene from bacteria, also degrading GABA, they can improve transformation in tomato and sweet cane. And then adding the ACDS and GABT expression cassettes onto the same plasmid and using both of those strategies at once gave pretty significant increases in transformation, which had an additive effect. This group called their strains carrying their construct here super Agrobacterium. Finally, salicylic acid, another plant hormone known to be involved in biotic stress response. This gene NAG can reduce salicylic acid to catechol. And initially it was shown that when you express this NAG gene in transgenic Arabidopsis and tomato lines, they become hypersusceptible to Agrobacterium transformation. And then pretty recently, last year, there was a paper published using a ternary vector that contained expression cassettes for ACDS, GABT, NAG, and then what's considered the constitutive always-on mutant of the VIR-G master regulator of virulence genes. And that also gave significant, I think, 3.5-fold increases in tomato transformation. Another kind of interesting thing I want to point out as far as NTEs is a whole gene module system used for host immune evasion. So this involved porting a type 3 secretion system, which ordinarily it's expressed in not only Pseudomonas, but several other plant pathogens and symbiotes. And typically, this secretion system transmits proteins from the bacterial cell into the host, usually for the purpose of suppressing plant immunity, which starts to get going in response to the plant's perception of bacterial signals. So these researchers showed that having a strain that houses a plasmid carrying all the machinery to make the type 3 secretion system, a plasmid expressing type 3 effector proteins that could be trafficked through that, a binary vector containing the tDNA, and actually not shown here, still a disarmed virulence plasmid. So technically this would be a quaternary system. They could get pretty substantial increases in transformation frequency over a binary alone, secretion system without effectors or an empty vector control. But I think what's actually more interesting about this is that they show that a plant-derived hormone or protein known to positively affect plant transformation, they could fuse this protein to a type 3 secretion N-terminal peptide, express it in Agrobacterium, have it trafficked through the T3SS into host plant cells, and also get really substantial increases in transformation frequency in monocot and dicot crops. So newer tools that have been published recently for accomplishing strain engineering. Kind of first, I'll talk about the classical method that's been used to engineer strains, which is called double crossover homologous recombination. So this necessitates using two sequences taken from the target strain, flanking the region to be deleted or replaced. We'll use tDNA in this case. And these are referred to as homology arms. They're usually 800 to 1200 base pairs long. Those are cloned into a disarmament construct, flanking usually a selectable marker. And then you have also a different selectable marker in the backbone. You introduce this construct into the target strain. Usually it is a non-replicating or suicide plasmid. So when you select for the marker in between the homology arms, what you should get are strains where recombination has taken place, which can occur at either homology arm. So what you first get is a co-integrate between your construct and the virulence plasmid. And then what you want to have happen is a second recombination event taking place between the other sequences of the other homology arm, resulting in a double crossover event that still contains your flanking selectable marker. Now, to identify these with this kind of setup, you had to screen for colonies that had a resistance to the antibiotic marker in between the homology arms, but susceptibility to the one in the backbone to ensure that that marker was absent and the target sequence was actually deleted. I want to talk about the gantry system, which Greg did describe already, so I don't want to go into too much detail here, but as he mentioned, this is kind of going backwards in biotechnology history to a unitary system where we have kind of a landing pad on a disarmed virulence plasmid where we can clone our gene of interest into what we call a donor plasmid, and then along with a helper plasmid that expresses recombinase proteins, we deliver both of these into the agrobacterium, and those recombinases facilitate loading of the cargo onto the tDNA. As Greg mentioned, we really like using this a lot in the laboratory because you can, with two different sets of donor and helper plasmids, you can do this iteratively, develop these really long tDNAs, which is necessary for Greg's work, and they're of an indeterminate size, really. So you're really limited by the efficiency of transgenes this long getting into plants. And I want to point out that, there's also other supposed benefits of using gantry over binaries, in that it's less likely to deliver multiple copies of transgenes, which is usually desirable for at least plants that might go into the field. And then you have less of a problem with backbone integration than you do with binaries. And I'll also say that I think a lot of people are intimidated by adopting this system because it's a lot more, it seems like a lot more labor, but I think with a couple tweaks to our kind of conventional approaches to how we deliver genes to Agrobacterium, it's not as scary as it looks, and it's pretty tractable. Now, as far as engineering genes in Agrobacterium, a CRISPR cytidine base editor was published in 2021 by a group, again, at KU Leuven and the University of Ghent in Belgium. And it consists of a catalytically inactivated Cas9 fused to a cytidine deaminase and a uracil glycosylase. So this whole chimeric protein is expressed under the control of a virulence gene inducible promoter, and then you have another cassette with constitutive expression of a guide RNA sequence. The construct also contains a SAC-B, which causes sucrose put in media to turn into a toxic product, allowing you to evict this plasmid from a strain after it's done its job. These researchers use this strategy to generate gene knockouts using early stop codon strategy in disarmed and wild type strains that we commonly use in transformation. And they determine kind of an optimal editing window, which is kind of at the -17 to -19 position relative to the PAM sequence, which this, since it's a cytidine base editor, will only convert a C to a T.
Chris, just to say, you're over your 20 minutes. I don't know how much more you've got to go.
Okay, yeah. Let me just kind of wrap up these couple systems here. So there was kind of an improvement to this system using chromogenic gene markers and making construct assembly a little bit more efficient. And then another tool here is the integrate system, which was pioneered actually by Keunsub's group at Iowa State. And they developed a construct design which includes this operon of Cas and transposon genes, where you clone in your CRISPR RNA as well as a transposon-based cargo with arms on it. And when you deliver this into the genes, or into the strain, it causes insertion of the cargo downstream from the CRISPR RNA's target site. And they additionally show that by using LOXP as the insertion cargo, they can disarm strains using this method. Finally, there's a recombinering system reported, which involves expression of phage proteins, and when you express those, you can introduce a linear fragment that should catalyze recombination with pretty short homology arm sequences. Now, I will say this is how it's supposed to work in theory, but after acquiring these constructs from this group, we were unable to get them to work, even repeating their exact strain and target. I don't think I have time to really go into kind of comparing these systems, but they all have kind of unique advantages and disadvantages to their use. Now in our lab, generating novel strains, my project is working on kind of a construct assembly system that only requires inputting homology arms from a target strain, which are amplified by PCR. And then these two homology arms are combined with these pre-existing plasmid tools that we're designing as well. And then Golden Gate assembly is used to assemble these 5 parts into a construct that is ready to introduce into Agrobacterium. And now this is based on kind of that more old school method, double crossover homologous recombination, which if you are interested in why, I can tell you more about the details of that. At the same time in our lab, there is an NSF project which Greg is involved in, and our PhD student Zach. They are taking wild type strains outfitted with a binary plasmid and doing kind of mass transformation screening in poplar trees, looking at interesting transformation phenotypes, including increases in marker gene expression. And then finally, we are combining these lines of research to start producing wild type strains that look promising in these initial screens. We've generated a few ones that look pretty promising, and I want to mention that we're open to collaboration, getting these strains tested for transformation in plants that we don't transform. So if you're interested in possibly collaborating, please contact me here. And then I'd like to thank our Strauss Lab members who worked directly on this project and others who gave a lot of support. And I want to thank USDA AFRI for postdoctoral fellowship funding. Sorry, I ran over. Thank you.
That's great. Thank you. That was a really good overview of all the different ways that you can try and improve Agrobacterium. So thank you. Okay, so we go over to the question and answer. So there are quite a lot of questions in the box. Greg has answered quite a lot already, brilliantly. So before you ask the question, please just look in the open and the answered section, because it might have been answered already. So Greg has done a great job in answering some of the more sort of specific technical questions in there. Okay, right. So let me go to the. So maybe the first question was for Keunsub. Two questions that related really. Basically, one about, have you had an issue with ruby affecting plant growth and regeneration? And also somebody else asked about, has it affected sterility? There's been evidence in tomato and Arabidopsis, and whether you'd seen the same in monocots.
Yes, we also observed similar things. So in maize, if you see the very high level of pigmentation, they don't grow to the full maturity, right? Sometimes we have a perfect purple, homogeneously purple plant with a really nice set of a seed. However, it's rare. So and in our case, we use the 35S, double 35S promoter. But recently I realized that there was a enhancer duplication. So instead of a single enhancer right now, it has a triple enhancer. I'm not sure if that made the changes, but I think I was lucky enough and get it. And I have a single 35S promote driving ruby, exactly same thing, but with only single enhancer element. It was almost as high as the double enhancer version. So the thing is, if it's too strong, you don't have a fertile plant. We observe the same thing in soybean, and the sorghum is similar. If the tissue produces a lot of pigmentations, they don't regenerate.
Great, thank you.
Mm-hmm.
I think there was just another couple on ruby. I think actually you've probably answered that. What somebody asked about, why there's ruby expression in the root but not in the shoot. But you're saying sometimes you do find it in the shoot. It's just about the different levels.
Yeah, exactly.
Yeah.
Okay.
So then somebody's asked about where to find your protocols. I presume they'll be in the references that you've got in your talk, and that the recording is available, so people can look in the talk for the references. Is that
Yeah, that's correct. But we also have a, I didn't cite in this presentation, we have a method paper published using MPT2 as a selection, which is called the Spring Harbor Lab protocol. You can just search my name and then should be able to find the, paper, I hope.
Great, so that'd be useful with a
Yeah.
methods paper. Yes, again, more about Ruby here. Somebody said that they have been using Ruby and that they see pink and then the color goes, but the gene is still there. What could be the reason?
Likely silencing. It's a viral promoter, so it can be silenced.
Exactly. And then somebody's asking, have you looked at the copy number in intrastate plants with the ternary versus, say, the superbinary system?
I haven't made a side-by-side comparison with a superbinary vector, but we didn't see, compared to our binary vector system, and the copy number was similar. On average, about 3 copies per plant, and then around the 10 to 30% of them were single copy event. So it's not really huge deviation, but I was expecting much higher copy number average, but I guess if you have too many copy number integration, maybe it knock out some essential genes. Maybe they don't regenerate. Maybe that's why. But in terms of a copy number, we did not see huge bump.
And are those, when you say different copy numbers in different locations, do you also see more than one copy going in at the same location?
You can, yeah, I have seen that too. So some location, there were tandem copies integrated, but in other cases you see single copy integration in one place. But around the average, about 20%, 25% were a single copy event. Yeah.
Right.
So
yeah. So go on.
Okay.
Okay, and then there's another question around why do you think you get 0 transformation efficiency when you don't use a helper plasmid?
I think 2 reasons. First, our original protocol, we induced the virulence gene for several hours before you do the agro infection. And then the co-cultivation was 3 to 4 days. With a new method, we don't induce vir genes for several hours. We suspend the agro in an infection medium, which has a serocidin gone. And in fact, just submerge them for 5 minutes. And then co-cultivation is only overnight, up to 20 hours. So if we are interested in increasing the transformation frequency, we can extend the co-cultivation time. That is another factor. Or you can also increase the agro cell density, right? And you remember other protocol, different crops, you may incubate with the agro for not like 5 minutes, maybe 30 minutes, 1 hour, or 2 hours. So there are many different parameters we can optimize. But in our case, it worked so well, we didn't bother to optimize those parameters.
But I guess this is the sort of thing that comes out from all your talks in a way is, there's so many things you can try. If you've got a species that you just want to get an edited plant, I guess are the key things that you would start with?
Yeah, you need to understand what's the bottleneck in your system. So for monocots, because they are not natural host of Agrobacterium, in many cases, boosting vir genes is sufficient to overcome some of the bottlenecks. But if you observe some of the defense responses, I think Greg or Chris already talked about it, implementing the type 3 secretion system with the AVR-PTO, for instance, you can dampen the host defense. Or you can also think of implementing elongation factor, which can trigger PTI in some plant species. If you change it, replace it with some pseudomonas, which is like stealth EF, you can minimize, triggering host defense. So there are many, many ways we can engineer Agrobacterium. The possibilities there, but I found that the most time-consuming part is actually validating Agrobacterium strains. We can quickly generate engineered agro strain, but you have to prove them they are really good for plant transformation. And for different species, it takes very, very long time.
Oh, that's really, really helpful. Thank you. Somebody's asked, I presume that's you again.
That's a good question. So it's related to regeneration. So even if a B104 and B73 are their sister lines, B73 is not regenerable using standard protocol. And when we use the B104 protocol, we get nothing. Okay, and then we have to boost the regeneration, and we use it using non-integrating Wushel or altruistic Wushel approach. Then you can transform B73 using this protocol.
Great. Greg, maybe I could ask you, and this might be me that I didn't catch this, but in terms of sort of the hairy root transformation process and then regenerating a plant, are you doing that because those tree species are recalcitrant to callus sort of generation directly, or because it's quicker, or because like Keunsub's embryo sort of method versus callus?
Right. There's a lot of different developmental pathways you can go through. And I think for many of these really difficult species, taking a fully differentiated cell and having it form roots is just an easier developmental pathway than some of the alternatives like somatic embryogenesis or callus to shoot or other pathways like that. So our general strategy, with this approach is just you generate a whole lot of already transgenic tissue. In some ways, it's kind of similar to like callus culture in that way or something like that. And then hopefully we can make the shoot process easier by kind of forcing the amount of transgenic cells that are input. And a lot of these species actually sucker quite a bit, forming shoots from roots. So there is kind of a natural regeneration pathway in some of these species. So we've just found that, and monocots, of course, are totally different, that these woody dicots just require thinking in a different way. And because these hairy root culture techniques are kind of already used in a lot of these species, we hope that that technical expertise allows people in the public sector to run with it.
And I guess, as you say, in a way, then you're putting all your effort into regenerating something that is transformed.
Yeah, and you can even pre-select which ones have the exact edits you want. So you could propagate that as a clone. We've been able to do it in liquid culture. You could also move them ex vitro in a greenhouse, although it'd be difficult to then regenerate those shoots from that. But there's a number of different ways you could take it. Yeah.
Great. And there's actually a couple of questions for you as well in the Q&A.
Oh, wow.
So one says, have you tried using silencing suppressors such as P19 to increase sort of editing efficiency, I guess? And there's some thoughts on eucalyptus.
Yes. So thank you for the question, Vitor. So we have tried P19. If you have it expressed under a constitutive promoter, it really reduces the shoot regeneration response, which is something we're surprised about. Again, Cree is a pain in the butt. And so P19 was kind of low-hanging fruit for us to reduce that silencing. So P19, of course, is a silencing suppressor that you might use. We need it as part of the same transgene because if we're going to wait a month or 2 months before we trigger the excision, it would need to be expressed in that way. So we are actually trying inducible expression of P19 to see if that'll help, because excision right now seems like the bottleneck. And then I see transgene-free co-editing in eucalyptus. So that best performing genotype I showed you has about a 4% transformation rate. And for almost all of the rest, it's just 0. And the transgene-free kind of co-editing type of approaches, typically most of what you regenerate will be transgenic. Basically, you will insert a transgene. You're relying on that transient delivery of Agrobacterium to hopefully not integrate editing transgene. And so for these really difficult species, you're never going to get the 100 transgenic events you need to find that 2% that are transgene free. And so that, I think, is the real challenge. If you're a company, maybe you can do that and do that approach. But most of the stakeholders that I'm trying to build these for, it's just at present not efficient enough to be totally transgene free. So that I think is the bigger challenge. I would also challenge you to, transgenes are powerful and they're useful for biotechnology. And so I don't want to limit ourselves just to think about simple gene edits as the only path forward because there's a regulatory window. So please, as biotechnologists, think about transgenes too.
Great, thank you. And then there's the next question. I'm not sure I quite understand it, but it says, and whether this is Chris's expertise, do we have any idea of how these plasmids, apart from the TIRI ones, affect virulence and plant transformation?
I can answer that. So
Well done.
I think the challenge is that people historically have done surveys of Agrobacterium strains. For instance, like the number of galls if you inoculate a plant on its stem. And the virulence is going to be confounded with the regeneration effects of the native tDNA. And so some strains might be forming galls because the tDNA genes are really effective in that species, and some might be just because the tDNA delivery is good on that host. So they are confounded, and really these kind of engineering approaches are the first time where we can really tit for tat look at those interactions. And so I think it's an important question because one thing in the field is like, our agrobacterium evolved towards specific hosts. We often use the term hypervirulent to describe ones that work across species, but are there generalists and then specialists? We don't understand that well enough, I think.
I read the question. That question was from Zach, right?
Yeah.
I read it as being asking about accessory plasmids particularly. So as far as I know, I don't know of any genes on accessory plasmids that have been identified as being important for transformation. That may not be the case. I would expect if they are, this would be an older agrobacterium literature. But we do know that either removing the AT plasmid from C58, or we just saw it the European Agrobacterium Conference a couple weeks ago, that if you have a deletion in the AT plasmid, which often happens spontaneously, that it has lower gall induction. So there probably are factors that influence transformation. I just don't know if we've really drilled down on those.
Great, thank you. Then there's a couple of questions which are basically about effectively how big your vector is and what the impact of that on transformation efficiency. I don't know, Keunsub, is that one for you?
Yeah. So you mean the tDNA part of your binary vector?
Yeah, the bit that actually is moving, I think. Yeah.
Okay. So there is some effect. My typical tDNA is around from 10 kb to 27-ish kb. So with a vector over 25 kb or 30 kb, I found its agrobacterium transformation efficiency is lower. There's a kind of a limit about around the 30K, because suddenly I get fewer cells, colonies formed on the plate. But in terms of a transformation frequency, I have not done any specific comparison, side-by-side comparison with the vector size, but you can think of this way. Agrobacterium deliver tDNA from right border to the left border. And then there was a report from, I think, a USDA lab. I forgot. I think the Dr. Rosa Timoni mentioned about it at the SIBB meeting, that if you put different markers and then in the middle of the tDNA, and then screen the transgenic plant for the presence of each marker. So if you put the marker at the end of the tDNA, you have a less frequency. Because some transgenic plant only have the right border region, not the end of the left border. That's why it's important to put the selector marker gene near the left border, right? To ensure that everything is, the entire tDNA is transferred to the plant. Yes, there is some effect.
Great, thank you. Somebody's put a question about high 2A times B protocol. Maybe, I don't know, yeah, you can answer that.
as long as your genotype is regenerable with a standard tissue culture process, you can use this standard rapid method. It'll work as long as you can regenerate plant, right? Ternary helper system can enhance the TDNA delivery, so you can get the plants. Great.
There's a question about removing the marker gene after hairy roots. I mean, I guess, Greg, that's basically about, well, either use excision if you can't go through that sort of seed cycle, crossing cycle. I don't know if you've got anything you want to add to that question.
Yeah, I think this system as it is, you could just position the lock sites so that you keep the role genes for metabolite culture, for instance, and you retain your other gene of interest, and then you remove the selectable marker along with that process, and it would all pretty much work the same. So yeah, or you could remove the role genes like I showed in the presentation. So that flexibility is there.
There are also some applications of for plant engineering where they actually want to leave the role genes behind because usually for horticultural crops, it's just creating diversity, the weird architectural phenotypes that they cause. And it's,
Yeah.
I believe, generally regulated as non-GMO.
Yeah, it depends on the country, but yeah.
Right. And there's another question, Greg, about have you tried other recombinases? Yeah.
Yeah, so we have Flip and Phyc31, and in the past we've worked with a few others. We've mostly stuck with Cree because it's the most commonly used in plants, but you know, if you look in the literature, it's like this is a day-to-day tool in most animal labs, and almost no one uses it in plants. And so I think there's just lots of hiccups to using these, and they're nowhere near as universal as CRISPR. So we have tried Flip, but not with this specific transgene. Piggyback, I have seen a few papers on that. That's a transposase, so it's technically footprint free. So it would be, I think, it's been used in rice and maybe banana as well to generate some transgene-free material. So, you know, I think all of those strategies could work. Whether they would still have this silencing challenge, I'm not sure. I think one problem is that most people who use recombinases in plants are using it in a crossing strategy. So you have a constitutive Cree overexpressor and you cross it to remove the antibiotic marker in another line, and then use the progeny as a way to sort that out. And when in these clonal plants, we have to deliver it in the same transgene. So it's delivered in cis, and that creates all kinds of problems. And so I suspect, yeah, this native silencing response is maybe related to like a similar thing to an active transposase being triggered for gene silencing or something like that. So we just need to overcome those, and we are pretty focused on either other recombinases or ways to ameliorate that response during regeneration.
Right. I think there's no more questions at the moment.
I have a couple of questions for Keunsub if we're
okay, go ahead, Chris. He's a captive audience, yeah.
So it's my understanding that all the ternary vectors that are out there have their vir genes taken from the hypervirulent BO542 plasmid background. So one question I had was using the ternary vectors in strain backgrounds that have those vir genes in their disarmed backbone, would that lead to the same gains, which you did show that in EHA 105, it has that, right? Have you checked whether, since it's identical or nearly identical gene sequences, do you get homologous recombination between the ternary vector and the disarmed virulence?
Surprisingly, no, I have not. I always check when I introduce the helper plasmid. I check the integrity from the cell, and then I haven't seen a problem. I have seen occasional transposon insertion from LBF44.4. So if we are using LBF44.4 with the TN904 transposon, which was initially used to engineer the LBF44.4 strain, it can jump around. It can jump to RK2 backbone or PBS1 backbone. If you analyze your binary vector, this is not super common, but you may sometimes see the transposon tracking the tDNA into the plant. So don't get surprised. If you're using LBF44 based strain, you may see some transposon sequence into your tDNA. So now there's a new strain, new engineered strain Corteva made by deleting the transposons. So you can avoid those problems. And then sometimes it can be very, very tricky. Sometimes it's just, you can just ignore. You can just select the one event with the clean insertions.
Wow,
Okay.
that's really interesting. I'd never thought about
Yes.
that being a problem with that strain background.
Yeah.
My other question was, have you thought about adding to ternary vectors the chromosomal virulence CHV genes, and do you think that would have a major effect?
That is a great question. At some point I thought about it, because if you want to use a non-agrobacterium for transformation, right, such as Ensifer, for instance,
Yeah.
they may not have the SHEV-A, SHEV-I, SHEV-G, the system to sense the acidic pH, right? But I don't have a manpower to actually do the experiment. Yeah, but I think that's a good way. Yeah.
We've got a couple more questions.
Mhm.
I guess maybe the first one, Keunsub or Greg or Chris. What are your it's saying there most of the approaches depend on tissue culture. What are your thoughts on tissue culture free genome editing?
So I will mostly vouch in the negative on this. I know there's been a lot of work on this recently. So number one, I'm coming at this from a tree background, and just doing one experiment in the greenhouse without tissue culture, it takes up half of a greenhouse. And it's easier to manipulate a whole bunch of plants in vitro than it is in a greenhouse. It might be less expensive per experiment than soil, but it's just really quite unexpectedly challenging to monitor and prune and do all the horticultural things that you need to for that type of gene editing. In addition, there's been some movement on this employing viruses in order to deliver CRISPR-Cas reagents, and none of the off-the-shelf, well-studied ones move in our trees. So we've tested TRV, TMV, TSWV. I'm trying to remember some of the PVX and a few others, and none of them move in tree species. And so if you work in the Solanaceae, it's going to be great. But we just our virology of what tools we have and what's stable in Agrobacterium as a delivery reagent for those is a challenge in a lot of these non-model systems. So in terms of greenhouse stuff, we've just found it difficult. We've done quite a few experiments using kind of agromorphogens in the greenhouse, and it's been more difficult than we thought it would be. So could be a game changer, but we have fallen back on in vitro methods.
Yeah, I would agree. I think it's very attractive, but it very much depends on your plant species you're working on. What are the relative efficiencies as far as just like the labor involved of doing in vitro work versus in planta work? I think overall before in planta becomes kind of a widely applicable method, we just need a better way to positively select for transformed cells in a greenhouse environment. It's much easier to apply selection in tissue culture than it is in a greenhouse environment when you're dealing with whole plants. And negative selection doesn't seem like a great option because the vast majority of your cells are going to be non-transgenic. You're probably going to kill the whole plant if you're using composite plants.
Great, thank you. And then another last question at the moment is to Greg, I presume, is have you considered direct repeats at the end of your constructs to sort of engage with the SSA DNA repair pathway?
Yeah, so we haven't done too much work on this, although you'll notice in my constructs that there is a direct repeat on each end of the tDNA near the lock sites with a matrix attachment region. And so we're unsure at the moment whether that provides any benefit or not in terms of the excision process, it being a very similar sequence on each end. So I'm just not sure yet. It could be something worth testing. I think there's a second comment here about synthesizing up to 120 kb constructs. So if you're delivering with Agrobacterium, I do think that you will start to hit a wall past a certain point. And we've delivered successfully at pretty high rates stuff up to 60 and 70 kb, but I've never tested anything beyond that. And there are cases in the literature, for instance, in Arabidopsis floral dip, of constructs up to like 110-ish kb. So I do think, it's a long single-stranded DNA fragment, and I do think there are some physical limitations to that. So this dream of like a megabase delivery from Agrobacterium, I don't think we can achieve that. It's just not possible. I maybe I'm too much of a naysayer, but yeah,
I'll say
that's mine.
our collaborator, Roger Tillmany, who was involved in developing the gantry system. I don't want to speak too much on his behalf, but his idea is that about 80 kb, like you have diminishing returns the longer tDNA you have, but he thinks about 80 kb is kind of like the wall before it's just going to be too impractical for the amount of explains you would have to transform.
Great. Okay, so I think we've got all 30 questions answered, which has done a fantastic job, guys. Thank you very much. So I think if there's no more questions, it just leaves to me to really thank Isabel for organising, but also Chris, Keunsub and Greg for great presentations and really engaging with the questions and answers. I think I certainly found the presentations informative and useful, and you'll be able to access them recordings that Isabel will send the details of. And then I just wanted to also just remind you that to look out for the next webinar. So the next one that will be organised by the Global Plant Council will be for our sister journal, the Plant Biotechnology Journal, and then there'll be another plant journal one after that. And so thanks to everyone for your participation and for engaging, sending us lots of questions. Thank you.
Thank you, Katherine and Isabel.
Thank you so much.
Yeah,
Thank
thank
you.
Thanks, everyone.
Bye-bye.
Bye-bye.